Hi, this is Breanna,
Ivy, Hannah,
Isabelle,
and Vicky. This is MondoWorks Essential Videos number 1. Today we will be talking about PCR, or polymerase chain reaction.
developed by Carrie Mullis in 1985, PCR is the quickest and the more selective technique when preparing large amounts of a particular gene or a DNA sequence when the DNA source is impure or in small amounts. In most cases, DNA cloning would be more efficient and a better way, but in this particular case, PCR is a better choice. Any target segment can be amplified in vitro in a few hours. Each cycle of the PCR consists of a 3-step process, and with each cycle, there is a chain reaction that is produced that will eventually create an exponentially growing population of identical DNA molecules.
Following what Vicky said, we can see how the number of double-stranded copies of original DNA exponentially grows after each cycle, over the course of 30 cycles.
During the cycle, there are 4 requirements: a double-stranded DNA with a target sequence, a heat-resistant DNA polymerase, the 4 nucleotides of DNA, and 15 to 20 nucleotide DNA strands that will serve as primers. PCR needs a heat-resistant DNA polymerase enzyme to withstand the heat of the first cycle, eliminating the need to add DNA polymerases every time the DNA is denatured. The DNA polymerase enzyme most often used in PCR is the Taq polymerase, an enzyme isolated from the bacterium Thermus aquaticus, which is a thermophilic bacteria first discovered near Yellowstone National Park's dry seas. This bacterium can survive in near boiling temperatures and thrives at 72 degrees Celsius or 162 degrees Fahrenheit. One primer on the DNA strand should be complementary to one end of the target sequence on one strand, and a second primer should be complementary to the opposite end of the target sequence on the other strand.
So here we have a DNA strand. These are the non-target sequences, and this is the target sequence. The yellow here represents the sugar of the non-target sequence, and the white here represents the sugar of the target sequence. The black beads represent the phosphate groups of the sugar-phosphate backbone, and these are the bases. Green represents guanine, red represents cytosine, purple represents adenine, and blue represents thymine. Labeled here we have 5' end of the strand, and labeled here we have the 3' end of the opposite strand.
This is cycle 1 of PCR, or polymerase chain reaction. Cycle 1 yields 2 molecules. The first process of PCR is denaturation, where the DNA is heated briefly to separate the double-stranded DNA. The next process is called annealing, where the DNA strands are cooled to allow short-stranded DNA primers to form hydrogen bonds with the ends of target sequences on opposite strands. The DNA primers form a point where DNA polymerase can start replicating. In this model, the primers are shortened for simplicity, but in real life, the DNA primer should be at least 15 nucleotides long for specificity. The next process is called extension, where DNA polymerase adds nucleotides to the 3' end of each primer. In PCR, the most often DNA polymerase used is Taq polymerase, which extends the primers in a 5' to 3' direction. The end product of cycle 1 are 2 molecules that do not yet match the target sequence. And now we will begin cycle 2 of PCR, which yields 4 molecules. Just like in cycle 1, the first step of cycle 2 is denaturation, where the DNA is heated once again to separate the strands. Afterwards, the DNA is cooled and the DNA primers once again form complementary hydrogen bonds with the ends of target sequences on opposite strands. Afterwards, Taq polymerase begins extending the nucleotides in a 5' to 3' direction. At the end of PCR, 4 molecules are produced, but none of them match the target sequence yet. And now we begin cycle 3 of PCR, which yields 8 molecules. Once again, the first step is to heat the DNA molecules briefly to separate the double-stranded DNA. Afterwards, the DNA strands are cooled in order to allow the DNA primers to once again attach to opposite ends on opposite strands. polymerase then continues to extend the nucleotides in a 5' to 3' direction by adding bases to the 3' ends of the primers. At the end of the third cycle, 8 molecules of DNA are produced. 2 molecules, or a quarter of the molecules, match the target sequence exactly. Although for simplicity, we will not continue to further model PCR, the cycles can be repeated continuously to produce an exponentially growing population of DNA. After about 30 cycles, more than 99.99% of the molecules match the target sequence.
So now we will be talking about the speed and specificity of PCR. PCR only needs minute amounts of DNA, and DNA can be partially degraded as long as a few molecules have target sequence. The key to specificity is the primers, which hydrogen bond only to sequences at opposite ends of the target segment. The primers must be at least 15 nucleotides long. This can still not substitute for gene cloning in cells when large amounts of a gene are required. Occasional errors can limit the number of good copies made from PCR.
The discovery of PCR has had a major impact on the ways that biotechnology can be applied to various areas of research. For example, PCR can amplify DNA from tiny amounts of blood, tissue, or semen found at crime scenes. It can also be used for rapid parental diagnosis of genetic disorders in DNA of viral genes from cells infected with viruses that are difficult to detect, such as HIV. It can also allow medical scientists to diagnose hundreds of genetic disorders by using PCR with primers that target the genes associated with these disorders.
Among the genes for human diseases that have been identified are those for sickle cell disease, hemophilia, cystic fibrosis, Huntington's disease, and Duchenne muscular dystrophy. These diseases can be identified before the onset of symptoms, even before birth. PCR can also be used to identify symptomless carriers of potentially harmful recessive alleles, essentially replacing southern blotting for this purpose.
To conclude, each continuous 3-step cycle of PCR produces billions of copies of a target segment of DNA in a few hours. Polymerase chain reaction remains a huge part of biotechnology today and is used frequently in many real-life applications. So,
This is polymerase chain reaction, and we hope that's helpful.